Hello friends and my dear students, this is Kanita, serving as head in the Department of Biochemistry, JJ College of Arts and Science, Autonomous, Pudukkottai, Tamil Nadu, India. In this present video, we are going to see in detail about the mitochondria. The first observations of intracellular structures that probably represented mitochondria were published in the year 1894. Richard Almond in 1890s established them as cell organelles and called them bioblasts. The term mitochondria was coined by Carl Benta in the year 1898. Mitochondria are small, granular or filamentous bodies which are present in the cytoplasm of eukaryotic cell, and also they are known as powerhouse of the cell. Mitochondria generally are double membrane bound. They are energy converting organelles. They are present in all the eukaryotic cells. They are the sites of aerobic respiration. Mito means thread and chondrion means granule-like. As they are involved in the production of energy, they are also known as powerhouse of the cell. Mitochondria are semi-autonomous organelle because they have their own genetic material. There are two hypotheses about the origin of mitochondria: endosymbiotic and autogenous. The endosymbiotic hypothesis suggests that mitochondria were originally prokaryotic cells capable of implementing oxidative mechanisms that were not possible for eukaryotic cells. They became endosymbiotes living inside the eukaryote. In the autogenous hypothesis, mitochondria were born by splitting off a portion of DNA from the nucleus of the eukaryotic cell at the time of divergence with the prokaryotes. This DNA portion would have been enclosed by membranes which could not be crossed by proteins. Since mitochondria have many features in common with bacteria, the endosymbiotic hypothesis is more widely accepted. Here we could see few supports for the endosymbiotic theory. Here are few evidences for the endosymbiotic theory. The mitochondria are self-replicating like bacteria. They divide by binary fusion as the bacteria does. The inner membrane of the mitochondria is very much similar in composition to that of the membrane of bacteria. The DNA which is present in the mitochondria is structurally similar to the DNA which is seen in bacteria. The ribosomes, enzymes and transport systems are also similar to bacteria. The size of the mitochondria is also same as that of the bacteria. And the most important feature is the protein synthesis which is being carried out within the mitochondria is inhibited by the antibiotics which are able to inhibit the protein synthesis within the bacteria also. Now coming to the morphology of mitochondria. The size of the mitochondria ranges from 0.05 to 1 micrometer in diameter, whereas 1 to 10 micrometer in length. Generally, mitochondria are bean-shaped, but in few cases, we could see the elongated and thread-like mitochondria, for example, in fibroblasts. The number of mitochondria which are present in each cell varies from cell to cell. For example, in the rat liver, the number of mitochondria ranges to around 500, whereas in case of sea urchins, it ranges from 13,000 to 14,000. And generally, mitochondria are seen in those cells which require high level of energy. For example, sperm cells, muscles, etc. The mitochondrion is a semi-autonomous double membrane bound organelle found in most eukaryotic organisms. A mitochondrion has a double membrane. The inner one contains its chemiosmotic apparatus and has deep grooves which increases its surface area. While commonly depicted as an orange sausage with a blob inside of it, mitochondria can take many shapes and their intermembrane space is quite thin. A mitochondrion contains outer and inner membranes composed of phospholipid bilayers and proteins. The two membranes have different properties. Because of this double membraned organization, there are 5 distinct parts to a mitochondria. They are the outer mitochondrial membrane, the intermembrane space, the inner mitochondrial membrane, the cristae, and the matrix. The outer membrane of the mitochondria is very simple, which is a phospholipid bilayer. It is smooth in nature. It encloses the whole mitochondrion. It contains a protein called porins, and these porins allows the free passage for various molecules into the intermembrane space of the mitochondria. The inner mitochondrial membrane contains proteins with 3 types of functions. Those that perform the electron transport chain redox reactions, ATP synthase, which generates ATP in the matrix, specific transport proteins that regulate metabolite passage into and out of the mitochondrial matrix. Coming to a unique structure which is present in mitochondria, the cristae. The inner mitochondrial membrane is compartmentalized into numerous cristae which expand the surface area of the inner mitochondrial membrane, enhancing its ability to produce ATP. These folds are studded with small round bodies known as F1 particles or oxysomes. These are not simple random folds but rather invaginations of the inner membrane which can affect overall chemiosmotic function. The space which is present between the outer membrane and the inner membrane is known as the intermembrane space. This is also known as perimitochondrial space. Because the outer membrane is freely permeable to small molecules, the concentrations of small molecules such as ions and sugars in the intermembrane space is the same as in the cytosol. However, large proteins must have a specific signaling sequence to be transported across the outer membrane. So, the protein composition of this space is different from the protein composition of the cytosol. The matrix is a space enclosed by the inner membrane. It contains about 2/3 of the total proteins in a mitochondrion. The matrix is important in the production of ATP with the aid of the ATP synthase contained in the inner membrane. The matrix contains a highly concentrated mixture of hundreds of enzymes, special mitochondrial ribosomes, tRNA, and several copies of the mitochondrial DNA genome. Of the enzymes, the major functions include oxidation of pyruvate and fatty acids and the citric acid cycle. The DNA molecules are packaged into nucleoids by proteins. Mitochondria divide by binary fission, similar to bacterial cell division. The regulation of this division differs between eukaryotes. In many single-celled eukaryotes, their growth and divisions are linked to the cell cycle. For example, a single mitochondrion may divide synchronously with a nucleus. This division and segregation process must be tightly controlled so that each daughter cell receives at least one mitochondrion. In other eukaryotes, for example in mammals, mitochondria may replicate their DNA and divide mainly in response to the energy needs of the cell rather than in phase with the cell cycle. When the energy needs of a cell are high, mitochondria grows and divides. When the energy use is low, mitochondria are destroyed or become inactive. In such examples, and in contrast to the situation in many single-celled eukaryotes, mitochondria are apparently randomly distributed to the daughter cells during the division of the cytoplasm. Understanding of mitochondrial dynamics, which is described as the balance between mitochondrial fusion and fission, has revealed that functional and structural alterations in mitochondrial morphology are important factors in pathologies associated with several disease conditions. The mitochondria serves as a site of several metabolic reactions. In the outer membrane, we can see oxidation of epinephrine, degradation of tryptophan, elongation of fatty acid taking place. In inner membrane, oxidative phosphorylation takes place. In matrix, important metabolic reactions like Krebs cycle, beta oxidation, detoxification of ammonia in urea cycle, storage of calcium ions, etc. occurs. The mitochondrial genome is a circular DNA molecule of about 16 kilobases. It encodes 37 genes, 13 for subunits of respiratory complexes 1, 3, 4 and 5, 22 for mitochondrial tRNA and 2 for rRNA. One mitochondrion can contain 2 to 10 copies of its DNA. As in prokaryotes, there is a very high proportion of coding DNA and an absence of repeats. Mitochondrial genes are transcribed as multigenic transcripts which are cleaved and polyadenylated to yield mature mRNAs. Not all proteins necessary for mitochondrial function are encoded by the mitochondrial genome. Most are coded by genes in the cell nucleus, and the corresponding proteins are imported into the mitochondrion. Most mitochondrial genomes are circular, although exceptions have been reported. In general, mitochondrial DNA lacks introns, as is the case in the human mitochondrial genome. Between protein coding regions, tRNAs are present. During transcription, the tRNAs acquire their characteristic L shape that gets recognized and cleaved by specific enzymes. Mitochondrial tRNA genes have different sequences from the nuclear tRNAs, but looks-alikes of mitochondrial tRNAs have been found in the nuclear chromosomes with high sequence similarity. An individual's mitochondrial genes are not inherited by the same mechanism as nuclear genes. Typically, the mitochondria are inherited from one parent only. In humans, when an egg cell is fertilized by a sperm, the egg nucleus and sperm nucleus each contribute equally to the genetic makeup of the zygote nucleus. In contrast, the mitochondria and therefore the mitochondrial DNA usually come from the egg only. The sperm's mitochondria enter the egg but do not contribute genetic information to the embryo. Instead, paternal mitochondria are marked with ubiquitin to select them for later degradation inside the embryo. The egg cell contains relatively few mitochondria, but it is these mitochondria that survive and divide to populate the cells of the adult organism. Mitochondria are therefore, in most cases, inherited only from mothers, a pattern known as maternal inheritance. Coming to the functions of mitochondria, mitochondria is involved in carrying out various important functions like energy transducer of the cell. It is involved in Krebs cycle, electron transport chain, phosphorylation processes, which helps in the synthesis of ATP molecules. The ATP, that is the high energy molecules which are being synthesized, are also transported with the help of a transporter called adenine nucleotide translocase. We could see various enzymes which are required for the synthesis of lipids inside the mitochondria. Also, mitochondria is involved in the production of heat. One of the major functions of mitochondria is apoptosis, that is programmed cell death. Mitochondria contains few enzymes like caspases which help in bringing about programmed cell death. Mitochondria is also involved in the synthesis of estrogen and testosterone. It is also involved in carrying out the metabolic reactions related to neurotransmitters, cholesterol and heme synthesis. I hope all of you might be clear regarding the function and structure of mitochondria. Thank you.